Our next speaker is Soohong Min, and there we go. And he will be talking about a peptidergic pathway critical to satiety responses in Drosophila.
So, hello everyone. I would like to thank for the organizers and staff for giving me this opportunity to talk about my project. And as I said, I'm Soohong Min, currently 5th year PhD student from Jongkyung Chung's lab at Seoul National University. So today I'm going to talk about the identification of peptidergic pathway critical for satiety responses to regulate body weight in Drosophila. So, as you may know, body weight is critical for our life. So, for example, like the balance between body weight and health, once it's broken, then, for example, by body weight increase, you become posed at high risk of obesity, heart problems, hypertension, and stroke, and so on. So it's really important to know the body weight regulator. So it will be really tremendous for our healthcare. So to identify such body weight regulator, I performed a genetic screen using variety of neuropeptide GAP4 lines close to USTNT that blocks neuronal activity. So among 23 GAP4 driver lines screened, I found most of these lines show body weight decrease or not significant changes. But surprisingly, I found one line that showed striking body weight increase in both genders, called MIP-GAP4. By the way, MIP stands for myoinhibitory peptide. So the rest of my talk will be about MIP pathway in regulation of body weight. So when I silenced these MIP neurons, as you can see here, induced a dramatic body weight increase over 30% in both genders in this graph, as you can see. Also with a huge enlarged abdomen here. But this body weight increase was not due to any abdominal body growth during development. As you can see here, I compared nearly close to flies' height, and I don't see any difference between the flies with silenced MIP neurons and control flies. Also, although I didn't explain to you, but I did other specific inhibition of this neuron, and I still see the body weight increase. So why do these flies get obese? So one way I can think of was body weight could be affected mainly by food intake. So I try to provide the flies with a restricted feeding condition to see if the restricted feeding condition will rescue the body weight increase defect. So, and yes, you can see that the flies reared on ad libitum feeding condition still show body weight increase like here in both genders, but when I provide the flies with a restricted feeding, then the body weight increase was completely reversed to normal level. And this indicates that MIP neuron silencing mediated body weight increase was by food intake. So here I prepared a video showing that you see this fly with silenced MIP neurons eat forever compared to controls, as you can see. Also, I quantified 24-hour feeding of this fly, and you see huge increase in food intake. So how about activation of these neurons? Would I see body weight decrease or food intake? So to do that, I took advantage of thermogenetic activation protocol for behavior using TRIB-A1. So TRIB-A1 is a warmth-activated channel that excites neurons when exposed to temperature stimuli over 30 degrees Celsius. So using this protocol, I tried to activate this neuron, and this fly, with the activation of Min neurons, didn't need the food, even though he has been starved for 18 hours. Also, I monitored 24-hour feeding of this fly before activation and after activation and after termination of the neuronal activation. So before activation, you don't see any difference between the flies in food intake, but once the neuron is activated, you see marked reduced food intake here. But this reduced food intake was completely normalized up to the level or even higher than controls, probably by rebound feeding effect. So similarly, I saw reduced body weight when I activate these Min neurons, as you can see here in both genders. Also, this body weight decrease was normalized by termination of neurons. So having shown that manipulation of Min neurons affect the body weight and food intake, I tried to look at this MIP gene itself. So using highly specific MIP antibody, I could detect MIP expression throughout the MIP neurons in the CNS here. Also, I tried to knock down this MIP gene using this the MIP scaffold driver. You see mostly eliminated expression of MIP in the brain. Also, I observed that the knockdown of MIP gene in MIP neurons increased body weight. So to further strengthen my observation, I tried to generate a null mutation for MIP. So using homologous combination, here you can see entire MIP gene was replaced by mini white cassette. So to confirm that this is really null mutation, I used the antibody, MIP antibody, and stay in the brain that harbor 2 copies of MIP allele, and you see wiped out MIP expression in the brain. And also, I worried about any genetic background affecting the body weight phenotype, so I backcrossed this allele several times to Cantonese background. So using these mutants, I compare body weight and food intake here. So you see increased body weight compared to control in this mutant, but this increased body weight was completely normalized by genetic restoration of the MIP gene in this mutant background. Also, same for the food intake. You see marked increase in food intake, but this increased food intake was normalized by the Lesky experiment. So further, I asked whether MIP gene is required for the MIP neuron-mediated body weight control. One way I can do was to activate the MIP neurons in wild-type background or MIP mutant background. So as you can see here, you can compare these 2 bars. So when I activate these MIP neurons in wild-type background, you see marked decrease in body weight, but you don't see the decrease in this mutant background. So this suggests that MIP is indeed required for MIP neuron-mediated body control. But also further, I asked whether the sex peptide receptor, SPR, there is known only receptor for MIP so far. So I test this mutant, and you see still body weight decrease in this mutant background when I activate the MIP neurons compared to MIP mutant background. This suggests that this was particularly interesting for me because it suggests that there may be unknown receptor for MIP in terms of the body weight regulation. So to summarize my results so far, I showed you that silencing MIP neurons increased body weight through elevated food intake. And also I showed you that MIP neurons decrease, activation of MIP neurons decreased body weight through reduced food intake. Also I showed you that MIP is expressed in the CNS and required for body weight and food intake. Also, finally, I showed you that MIP is indeed required for MIP neuron-mediated body weight control and independent of SBR. So far, I showed you that manipulation of MIP neurons inhibits food intake and body weight. So how this happens? So I asked how this happens. So it could be that MIP pathway signals satiety to regulate body weight. So then how do I measure, quantify the abstract concept of the state in the flies? So I employed two behavioral paradigms to quantify satiety. So one is that the olfactory two-choice assay using a T-maze. So if flies are starved, and then they would be more attracted to food odor, but if they are not starved, satiated, then there would be less attracted to food odor. Also for taste, you see this fly extends his proboscis to sucrose, but if he is not starved, then his PR response is down. So using these two paradigms, I check their satiety level. So what I saw from these experiments was that if you silence these neurons, these flies more attracted to food odor, not only for yeast paste, but also for other fruity odors. Also for taste, you see increased level of PR to sucrose, as if they have been starved, even though they are not starved. So, however, when I activated these neurons, you see decreased sensitivity to food odor, also to sucrose here. So, taken all together, I'd like to propose a model in which MIP neurons induce satiety, thereby inhibit food intake, and ultimately body weight. So, but if MIP neurons activity is silenced, then satiety is not induced, so thereby food intake increased and eventually body weight. So in my last slide, I'd like to express my acknowledgement to my important collaborators, including my supervisor, Jong Kyung-chung, and thank you for your listening. I'll take any questions.
Questions?
Hi. I was wondering, in your females, when they get fat, are their ovaries getting fat, or is it their fat bodies?
Yeah. So I checked that, and the size of the ovaries were similar between control and the MIP neuron silenced flies.
Okay, cool, thanks.
Did you do all your experiments in a certain sex?
I'm sorry, I didn't hear you.
Did you use just males or just females for your behavior?
No, actually, I showed you both genders.
Okay.
So I did all the experiments both gender, using both genders.
Cool, thank you.
Okay, let's move on.